Thanks very much, Rupert. So this is moved from this morning. As you can see, this is a 20-minute talk now with a 30-minute slot. So I will ramble and digress a little bit, but I won't keep you from tea. And this is a little bit different from some of the other talks you've had today, because I'm going to talk really about a particular technology, not particularly related to a specific group of patients, although clearly where we're heading is for haemophilia. So this is about zinc finger nuclease therapy and haemophilia. And this, by way of introduction, allowing me to digress a little bit, this is just a timeline of treatment for haemophilia. And the reason I put it in is not because you need to know about the movement from cryoprecipitate through various forms of concentrate, recombinant concentrates and so on, through to gene therapy, which is what we're going to talk about, but because it looks like gene therapy stopped there. And there are many, many different forms of plasma and recombinant factor replacement therapy, and there's only one kind of gene therapy here. And in fact, Kathy Hyde, when she was talking this morning, pointed out that gene therapy seemed to have kind of settled on a particular technology. And I think it's actually not clear, I'm sure she would agree with me, it's not clear exactly what form gene therapy will be in the future, and it may not be the one that's being used in all the trials at the moment. It won't necessarily be zinc finger nuclease either. The other thing is that we glossed over a lot of the development work that went through for gene therapy of haemophilia. Many failed attempts made over quite a number of years. So ex vivo transduction of fibroblasts, taking fibroblasts, putting them back into the omentum or under the skin, direct infusion of retrovirus, infusion of gutless adenovirus, putting plasmid or virus directly into muscles, so multiple intramuscular injections of vector to try and get expression. Many of these produced transient expression, but it didn't last. And I think that the persistence of the therapeutic effect for gene therapy is obviously going to be one of these important issues for us in over, well, many years, maybe perhaps decades. I expect most of you will have seen several of these very successful reports of gene therapy for haemophilia in the last few years, all not surprisingly published in the New England Journal. So haemophilia B, this is the first one, 2011, the first real success using AAV, the adeno-associated virus, achieving expression of 2 to 11%, and importantly sustained, as far as we know, for all the years since, so six years or so since then, well, probably a bit longer for the patients who first went into the trial. This is Kathy Hyde's study with an engineered AAV, looking at activity here. So this is the Padua variant, so the activity is somewhat higher than the actual antigenic or molar concentration of the factor 9 produced, and so consequently getting higher levels. And the haemophilia A study was reported quite recently using AAV5, getting levels with admittedly substantially higher doses of the of numbers of viral genomes, but getting factor levels that are into the normal range. So this is pretty extraordinary, really. But all these gene therapies have a couple of things in common. One of them is they're somatic. So these are really only affecting the somatic cells. These are not germline changes. We're not really curing the patient, or to say we're not eliminating their haemophilia in quite a complete sense. Although, in fact, we're not sure we really want to get into that just at the moment, and somatic gene therapy is fine, but germ cell gene therapy, which would alter the transmission of the disease itself, is forbidden, basically, at the moment. It's too much of an unknown territory and potentially too dangerous and too close to things like eugenics, I think, for a lot of people to get into. The fact is, actually, though, we're not... the number of people with haemophilia is increasing. It must be increasing. I think it is increasing if you look at the data. We've removed, more or less, any selective pressure on the factor VIII gene. So a number of people with haemophilia will just go on increasing until, as Ted Tuddenham used to say to me, it reaches fixation. And everybody's accumulated over the many, many years a mutation in the factor VIII gene, and they'll quite happily be quite able to pass it on because we'll fix the problem with gene therapy or replacement therapy or something like that. So maybe we need some other tax as well somewhere along the line. The other point about the AAV technology that's been sort of established as the current standard for gene therapy trials, is that the AAV, the adeno-associated virus, is episomal. So what this means is, and I'm sorry if I teach any of you to suck eggs, I'm sure many of you know this, that the viral particle takes the gene cargo into the cell, and the gene remains in an episome, which is separate from the genome, it's separate from all the chromosomes. And I think Kathy Hyde touched on this as well. So that means it's not actually part of the replicative process that happens when the cell divides. So this means, as is just shown in the picture on the left here, that as the cells divide, then although the genome, the DNA in the cell that makes it a cell, is copied, replicated, and there's a copy for both of the daughter cells, that isn't true of the episome. The episome doesn't get copied, and as the cells divide and divide and divide, the number of episomes per cell will go down and down and down, and so it'll be lost. So when is that likely to be important? Well, it may be important in the long term, in the very long term, if you give it to someone who's 10 or 20 and they live till they're 80. If there's a regenerative process, I don't know the turnover in the livers, but it's a possible route for loss of the therapeutic episome that you've put in. So far, that doesn't seem to be a problem. So as I mentioned in the Factor 9 trial, 7 or so years later, the levels seem to be steady. And in the dogs, the same technology was used to treat dogs, and one dog lifetime, whatever that relevance that has, 10 or 12 years later, these were still had very steady levels all the way through. So it looks quite promising. But the other circumstance in which this might be problematic, if you want to treat young people whose livers have not grown, because if you give a dose of episomes to a child, maybe a very small child, then as they grow and their liver grows, then you'll have given them, still given them the same dose, but it'll now be an inadequate dose for the size they are. And that's why Kathy mentioned that maybe one approach to that would be to find a way of retreating them. So giving them a top-up dose of adenovirus so that they were able to make enough Factor 9 for their bigger body in their bigger liver. But another way of doing that, of course, would be to give them a therapy that actually integrated into their genome so that it was repeated, replicated as they grew. So this brings me to the topic of gene editing, genome editing. So what will this do? What we mean by doing this is that we're going to alter the existing genome. We're not just going to stick in something on top as extra sideshow for their ordinary genome. We're going to actually alter their genetic makeup. So why would that be useful? Well, it allows you, for one thing, to target the defective gene. And now that may be useful if you've got a disorder that's, say, a dominant negative disorder, and you actually want to disrupt it or replace it, something like that. You could also, if you want to simply add a gene, put it into a particular site in the genome. So the targets might be the gene that you're interested in, or it might be some other spot that you think is a good idea to aim for. The advantage of that would be that it's passed on to the progeny. So now it's in the genome, this new gene, or this corrected gene, will be copied either in toto or in its corrected form into the progeny of the cell, and it will be passed on. So as the liver grows, for example, so the number of copies would grow. So as I mentioned, this might be good for dominant negative disorders. The other thing is it allows you to utilize genomic regulatory elements. So you can imagine that if it's a particular gene that you want to be expressed in a particular way, you can either put in, and maybe you think of something like thalassemia, where the alpha-beta ratio is going to be important. You would put it either into the correct regulatory context in the cell, so it'd be alongside the things that normally regulated the way that gene is transcribed, the amount of which it's transcribed, or you could put in the regulatory elements with it. So for thalassemia, in some experimental models, the whole locus has been put in. So it goes along with its regulatory elements. Very difficult to do in an episome. But of course, that's not without problems. This is serious business, mucking about with somebody's genetic, core genetic material. So you'd want to make sure you're happy about doing this, I think you'd have to be happy that you had specificity, that you're only going to do this, make these changes, in the particular spot that you want to make it. You don't want off-target effects. So if you're trying to knock out a deleterious dominant negative gene, you don't want to be, by sort of putting a nonsense into it, you don't want your technology to go around putting nonsenses in other critical genes all over the shop. It would not be a good thing. Also, we're going to put in a mechanism into the cell which is capable of breaking DNA and repairing it and doing things like that. And we wanted to do it only once, really, in the cell. So you don't want this activity knocking around in the cell for a very long period of time, because firstly, you might ruin what you've already done, and secondly, you're inviting off-target effects by producing genetic changes in other places you don't want them. So what we need is nuclease activity to a specific genomic site. And now we're getting closer to the title of the talk. So one way of doing this, there are a number of ways of doing it. You may have heard of things called TALENs, or you may have heard of CRISPR. These are ways of doing that thing. This is actually called zinc finger nucleases. Right, so let's look at what this involves. Well, this involves putting into the cell, I'll come to how that's done in a moment, but it will use an adeno-associated virus, a protein that looks like this. This is a zinc finger protein. Now, you probably know about zinc finger proteins because they're ubiquitous DNA-binding proteins that are utilized by the cell for regulatory purposes. Often these are transcription factors, very often. So the zinc finger protein has a protein sequence that binds to a specific DNA sequence, and it's very specific. So that's one half of the thing we're going to put in. The other half is a nuclease. So this is like a restriction endonuclease that many of you have used, like BAM-H1 and HIND-3 and these sort of things. But this one is called FOC1, and it's a little bit unusual actually, and I was interested to find out about this. It actually has to dimerize to be active. Okay, so it's not going to be generally active unless it dimerizes. So what we're going to do is put one zinc finger protein with one half of the FOC1 here that binds to this specific sequence, and another one that binds to this specific sequence and carries the other half of the FOC1. So the specificity is guaranteed by the zinc finger binding to this particular DNA sequence, and the bigger we make this, then the more specific it becomes. And it will only be able to cleave the DNA when its counterpart is bound to this particular sequence just here. So I've got specificity for the whole of this sequence, and the FOC1 will make a cleave here. More than that, actually, which makes it very clever, is that once we've broken this and the two things have come apart, depending on what we do to them, then it can't work anymore because the two binding sites are now separate, and they can't bring the two FOC1 portions together. Right. So what this will do at a particular site of our choosing, if we're clever enough to make the protein appropriately, is introduce a double-stranded break. And this enzyme makes a rather odd break, and it's a unusually staggered. It's quite a complicated stagger, but it's a staggered break. But nonetheless, it's a double-stranded break in the DNA at this particular site. And actually, although this is jumping ahead slightly, this particular site that's been chosen for the study I'll talk about is in the albumin. Okay, it's in the intron, the first intron in the albumin gene. So what happens after you made a double-stranded break? So here's the here's that thing again. Here's the nuclease, and here's the FOC1. It makes a double-stranded break. Now these cells have quite double-stranded breaks are very dangerous for cells. So you actually have quite complicated and thorough mechanisms for repairing them. Obviously, it's difficult to repair them perfectly, but you have a number of different methods. One, you have what's called the homology-directed repair. So these are all repair mechanisms for double-stranded break. This goes on mostly in replicating cells. And if you think about it, there are a number of situations when cells need to be able to repair double-stranded breaks. So meiosis and crossover recombination is one of them. There's another one which goes on when you're making your immunoglobulin genes and you undergo VDJ, recombination events. That's actually a bit more complicated, and that's uses a form of non-homologous repair. So the short point is that the cells do have means of repairing the double-stranded break that you've made with your zinc finger nuclease and FOC1. And what we'd like it to do is utilize a particular bit of DNA that we're also going to put in the cell to make the repair. And we can do this by using homology arms, which also bind to these regions here. And if it uses this as a template, then it'll undergo homology-driven double-stranded repair. But that's not actually so common in the cells we're going to target, because that's mostly going on only in replicating cells. But the other thing that can happen is it can utilize this this segment we put in, use non-homologous end joining, and it'll stick this transgene in here. So you can imagine what's going to happen. You need 3 things to go in. You need this half, that half, and this template. And then what you're going to put a lot of these in, and what's essentially a relatively rare event will put the transgene into the hole you've made in the particular locus that you've targeted with your zinc finger nuclease. So as I mentioned, the target spot for this that's been chosen for these studies is the albumin locus. Now why? So there are several advantages. One is it's tissue specific, so the locus is only active transcriptionally in the liver, but in the liver it is incredibly active. So the albumin's a major, maybe 25% of the output of liver cells. So it has a powerful promoter. But the liver's so big and it makes so much albumin that these few rare events is not going to make someone hyperalbuminemic. And this technique is site-specific and it's lost after integration. So the site is here in the first intron in the albumin gene. And what we're going to do is put the transgene into that spot so that it is it's driven by the very powerful albumin promoter. It's only driven by the albumin promoter in the liver cells, for which the viruses are tropic anyway. And we can afford to down-regulate, lose a little bit of albumin production to make wafers and factor 9. So that may sound more or less incredible to you as a kind of speculative shot of several different bits of machinery into the cell. In fact, it works. So this is a murine experiment. What you do is you put in these the AAV8s with the zinc finger nucleases and the AAV8s with the factor 9 gene, and afterwards, quite remarkably, you get factor 9 expression, which is sustained, and not much effect on the ALT. So actually, it does work, rather remarkably, and it's sustained there. And this is more or less the same data. This is published in Blood, showing the factor 9, and perhaps what relevant for you here is the percent of normal, the antigen and activity. These are for mice, I should say. There's a lot of steps between murine, larger animals, and humans, obviously, because all the different steps are not necessarily as efficient in all those different molecules, all those different animals. But this, I think, is one of the most interesting things about this. So here's the experiment where the zinc finger nucleases and the factor 9 cargo, as it were, has been put in. And in this experiment over here, this is a non-integrating control. So this is what you might call a standard gene therapy episomal experiment. And then what's been done in this experiment is a partial hepatectomy is being performed. So most of the liver is chopped away. So what happens then? Well, the liver has to regenerate. And exactly the situation I was talking about before has to follow. All the liver cells have to replicate and divide, and they'll copy their cells. So in the zinc finger nuclease experiment, where the factor 9 donor, cDNA, has been put into the genome, following the partial hepatectomy, there's a bit of a bump up and down, but it it maintains production. In a non-integrated experiment, where the factor IX has come up, after the partial hepatectomy, the liver recovers, but there's not enough episomes there. So the factor IX production does not recover. So that's just quite a nice way of showing the difference in effect between integrative gene therapy and non-integrative. Not, of course, that most of your patients are going to be having partial hepatectomies, but they may, for example, in the future, be wanting to grow. Okay, so this is just a brief summary. So the murine data shows that you can get expression and it's sustained up to 60 weeks and it survives partial hepatectomy. And although this isn't published, the company I'll mention in a moment have non-human primates, so monkey data, showing also that you can get substantial expression and that sustained expression. One other thing I mentioned earlier on, you may still have in the back of your mind, is about off-target effects. Well, if you do a sort of genome analysis, you can look for areas in the genome that might be similar, might be just about sufficiently similar to the target in the albumin intron. But in fact, no off-target modification. You found 80 predicted possible off-target sites, and none of them actually were hit by this method. In cell-based experiments, there was one other locus that was found to be transformed at a much lower rate, about 30-fold lower rate than the target one in albumin. But in fact, in the animals, this wasn't targeted at all. It wasn't found to be targeted, so that's something of an in vitro effect. One general point about this that Sangamo have made to me is that this of course, could be used, as indeed could be episomal therapy, for a number of other things. So there'll be trials for not only haemophilia B, but for mucopolysaccharidoses and Gaucher's and Fabry's as well, which might be interesting. I know some departments of haematology look after these people. So very briefly, just to finish off, to mention there will be a study using this technology to attempt to correct factor IX deficiency in people with haemophilia. And it will be a dose escalation study targeting the albumin locus. A small number of people have been screened already in the US. Similar things will apply to the other studies. So this is the zinc finger nuclease and the target are got into the cell using adenovirus capsid. So it's still an antibody screening step necessary during enrollment for the study. And it will be interesting. Of course, this is, you'll realise it's unknown. We don't know quite what the efficacy will be as you go from mouse to monkey to human. But it is another way of achieving gene therapy. Thanks.